Dear students, in this video I am going to explain about glycosides based on PCI syllabus 5th semester Pharmacognosy and Phytochemistry-II. In this unit we have three crude drugs: Senna, Aloe, bitter almond. So coming to the definition of glycosides, these are organic compound obtained from plants and animals which on acid or enzymatic hydrolysis gives sugar and non-sugar portion. This sugar portion is also known as glycone, and this non-sugar portion is also known as aglycone or genin. This glycone and aglycone portions are connected by a linkage known as glycosidic linkage. Some examples for glycones are glucose, fructose, digitoxose, galactose, mannose, etc. Example for aglycone portions are alcohol, phenol, anthraquinone, amines, etc. These glycosides are occur in various parts of plants like fruits, seeds, leaves, barks, etc. Next is properties of glycosides. These are colorless, crystalline or amorphous in nature, water and alcohol soluble. Aglycone portion is soluble in benzene, either etc. like organic solvents. Then their activity, therapeutic activity is based on mainly due to the presence of aglycone part. Then general functions of glycosides mainly involved in growth regulation, protection, energy source, pollination, then some disease like antibacterial activity, etc. Chemically, they are stored in inactive form. On enzymatic hydrolysis, they will convert it or they will activate it. Next is classification of glycosides. We can divide glycosides into four based on chemical nature of aglycone moiety, based on presence of glycone portion, based on linkage between glycone and aglycone portion, and based on therapeutic activity. So first is based on chemical nature of aglycone. We have different types of glycosides according to this. One is anthraquinone or anthracene glycoside, sterol or cardiac glycoside, saponin glycoside, alcohol glycoside, phenol, coumarin or furanocoumarin, isothiocyanate, cyanogenetic, flavanol, iridoid glycoside, and glycosidyl bitters. So first one is anthraquinone or anthracene glycoside. This is the structure of anthraquinone ring. Here the aglycone portion is anthraquinone. So there are different examples like senoside in senna, aloe in aloe, rene in rhubarb, etc. Next is steroidal glycosides. This is the structure of steroidal ring. Here the aglycone portion is steroidal ring based on carbon 23 or carbon 25 containing steroids with 5 or 6 membered lactone ring. If it is 5 membered lactone ring is attached, it is known as cardinolides. If it is 6 membered lactone ring is attached, it is known as bufadinolides. Some example for steroidal glycosides are digitalis, then tevacia, that is tevatin, then mainly used for cardiac action. Next is saponin glycosides. Here the aglycone portion is sapogenins. These toxic sapogenins are known as sapotoxins. And, some example for sapogenin glycosides are diosgenin, that is Dioscorea drug, then glycyrrhizin, licorice. Next is alcohol glycosides. Here the aglycone portion is alcohol used for antipyretic, anti-inflammatory actions. Examples are salicin. Next is phenol glycoside. Here the glycone portion is phenol. Example is bearberry. The next is coumarin or furanocoumarin glycosides. Example is Soralia, that is Soralen. Some structures I have given here: coumarin and furanocoumarin, basic nucleus. So seventh one is isothiocyanate glycosides. Here the aglycone portion is isothiocyanate. Examples are cinegrin from black mustard, then cinalbin from white mustard, etc. Then next is cyanogenetic glycoside. We have one drug for your this semester, that is bitter almond under cyanogenetic glycoside. So some examples: bitter almond, it contains amygdalin. Other examples are wild cherry bark. Then next is flavanol glycosides. Here the glycone portion is flavanol. Examples are rutin, quercetin, etc. Here I have given one example also. The next is iridoid glycosides. Here the aglycone portion is iridoid structure. It's a type of monoterpenoids. Example is catalpin. Then Next is glycosidyl bitters. Examples are gentian. Then so this is the concise table for all the glycosides and examples. So in this we have Aloe, we need to discuss, then Senna, then bitter almond. So these three drugs are included in your syllabus. So that in this video I am going to explain these three drugs, its biological source, its uses, chemical constant, etc. So next is the based on presence of glycone portion. The based on sugar portion, we have whether it is the sugar is glucose, that name is glucoside. Whether it is fructose, it is known as fructoside, pentose, pentoside, etc. Third classification is based on linkage between glycone and aglycone portion. So first glycosidic linkage is C-glycosidic linkage. So C-glycoside. So here the hydroxyl group of glycone is react with CH group of aglycone portion, forms a glycone C linkage aglycone with the removal of water. So some example for C-glycosides are aloe in aloe, cascaroside in cascara, carbonic acid in cochineal. It's an insect, animal source of drug. Next linkage is O-glycoside. So here the hydroxyl group of glycone is react with the OH group of aglycone portion. With the elimination of water, it will form glycone or aglycone linkage. Examples are sennoside in senna, rain in rhubarb. The next linkage is S-glycoside or S-glycosidic linkage. Here the hydroxyl group of glycone is react with the SH group of aglycone. It forms glycone S-aglycone with the elimination of water. Some examples are sinigrin in mustard. Then N-glycoside linkage. Here the hydroxyl group of glycone portion is reactive with NH group of aglycone portion. With the elimination of water, it will form glycone and aglycone. Some examples are nucleosides like DNA and RNA. The last classification on the basis of therapeutic activity. So the pharmacological action based classification. Some examples: cardiotonic means example digitalis stevia, anti-diabetic gymnema, analgesics and anti-inflammatory licorice, purgative, aloe senna, anti-ulcer licorice, expectorant squill, stomachic gentian. So general chemical test for next is general chemical test for glycosides. So first we have to prepare one powder drug of glycoside with treated with sulfuric acid and NaOH 5% NaOH solution. The mixture is to the mixture we need to add fillings A and B. It forms some red coloration. It indicates the presence of glycosides. There are some specific tests for glycosides. One is test for anthraquinone glycosides. Some name reactions like Bontrager's test. Specific test for anthraquinone glycoside is one is Bontrager's test, another one is modified Bontrager's test. Bontrager's test, the drug is boiled with sulfuric acid filter. To the filtrate, we need to add any organic solvents like benzene or chloroform or carbon tetrachloride. Then organic layer we need to separate and it is treated with ammonia solution. The ammonia layer become pink color to pink to red color. It indicates the presence of all glycosides. The next is modified Bontrager's test. Similar to Bontrager's only exception is the drug is boiled with dilute HCl, dilute sulfuric acid and ferric chloride. Then we need to filter. To the filtrate again we need to add organic solvents like benzene or chloroform. Then shake well. Organic layer is separated. Again we need to treat ammonia solution. The ammonia layer become pink to red color because of the presence of C-glycosides. Next is test for cardiac glycoside. We have different name reactions like Legal's test to the alcoholic solution of drug which we need to treat with pyridine and sodium nitroprusside. It forms pink to red color. It indicates the presence of cardiac glycoside. Next is Baljet test. Alcoholic solution of drug is treated with sodium picrite solution. Yellow to orange color it forms, indicate the presence of cardiac glycoside. Next is Keller-Kintiani test, important test. We have to boil 1 gram of finely powdered glycoside drug with a 10 ml 70% alcohol solution for 2 to 3 minutes. Then filter. To the filtrate we need to add 5 ml water and 0.5 ml strong lead acetate solution. Shake well and separate the filtrate. To the filtrate, we need to add equal volume of chloroform and evaporate to yield. It gives some extractives. So that extractives we need to treat with glacial acetic acid and then cool. Then add some drops of ferric chloride solution, and it is transferred to the contents is transferred into the test tube containing 2 ml concentrated sulfuric acid. So the reddish brown layer changes to bluish green. It indicates the presence of cardiac glycosides. Next test is test for cyanogenetic glycoside. Sodium picrate test. Here the aqueous solution of drug is treated with the dilute sulfuric acid, placed in a flask and suspend sodium nitrate filter paper to it. So the filter paper turns brick red, indicate the presence of cyanogenetic glycosides. Next test is test for saponin glycosides. Two tests are there. One is form test and the one is hemolysis test. So in form test, the powder drug is treated with water, shaken well. It forms stable froth or foam. It indicates the presence of saponin glycosides. Next is hemolysis test. Here the powder drug is treated with 0.2 ml blood in normal saline solution. Hemolysis occurs means indicate the presence of saponin glycosides. Next is test for flavonoid glycoside. First one is Shinoda test. Here the alcoholic solution of drug is treated with hydrochloric acid and 0.5 gram magnesium tonics. It indicates the presence of pink color, indicate the presence of flavonoid glycosides. Next is alkali test. Here the alcoholic solution of drug is treated with sodium hydroxide solution. That is one drug is treated with alkali solution. The yellow color which disappears on treating with the acid, it indicates the presence of flavonoid glycosides. Then one more test, lead acetate test. Here the powder drug is treated with the lead acetate solution. Yellow precipitate forms indicate the presence of flavonoid glycosides. Then next is coumarin glycosides. Alkali test. Here the powder drug is treated with alcohol and caustic soda. The bluish green fluorescence occurs, indicate the presence of coumarin glycosides. Already it's there in your fourth semester cognosy in detail, but here, so in your syllabus we have to concentrate on a few crude drugs. That is, one is cyanogenetic glycosides. Under this we have bitter almond. Then one more category is there, that is anthraquinone glycoside. We have two drugs, Senna and Aloe. So in case of bitter almond, we need to discuss biological source, synonym, microscopical characters, chemical constituents, uses, etc. So synonym for bitter almond, it is amygdala amara and badam. Then biological source, it consists of dried ripe seeds of Prunus amygdalus, belong to the family Rosaceae family. Then geographical source, it is cultivated in Italy, Spain, Portugal, etc. They are native of Iran, Asia, etc. So before we know that sweet almond, right? So this is bitter almond. Prunus amygdalus belong to family Rosaceae. Macroscopical characters are color brown, order orderless, taste bitter, size 20 millimeter length, 125 millimeter width, then 10 millimeter thickness, then shape flat, oblong, ovoid. Then solubility is, it is insoluble in ether and soluble in water. It is soluble in water. Next is chemical constituent of bitter almond. So mainly already we are discussing about glycosides. So first we need to mention it is glycoside. Then which type of glycoside? It is cyanogenetic glycoside. Then chiefly it contains a bitter glycoside known as amygdalin. So bitter almond contain amygdalin. Then other constants are 40 to 50% fixed oils, 20% protein, 0.5% volatile oil, etc. Then this amygdalin on hydrolysis gives benzaldehyde and hydrocyanic acid. This is the structure of amygdalin. That is the chief constant of bitter almond. Then these are the uses of bitter almond. Due to the presence of hydrocyanic acid, these are used as sedative. Then some fixed oil of bitter almond is used as demulcent in skin lotion, soothing property. Then it is also used in form of liquors, some bitter almond water, etc. Second category is anthraquinone glycoside. Here we have two crude drugs we need to explain. One is Aloe, one is Senna. So coming to the Senna, the synonym is Kumari Muzaba. Biological source, it consists of dried juice of leaves of Aloe barbadensis, that is Curacao aloe, Aloe perry, Socotra aloe or Aloe ferox, Zanzibar aloe, and other hybrids of the species with Aloe africana, that is Cape aloe, and Aloe spicata belong to the family Liliaceae. So these Curacao, Zanzibar, then Cape, all these are some place names. So based on this place name, they are known as, this genus is known as these place names only. The geographical source means it is indigenous to some southern Africa, Cape, Zanzibar, islands of Scotland, etc. Also cultivated in Caribbean island, Europe, etc. Next is description and microscopy. So different types of aloe is having different color, different texture, etc. So Cape aloe, color is dark brown or greenish brown in color, glossy mass, transparent appearance. Then in case of Curacao aloe, its color is dark chocolate brown and fracture is waxy. Then Socotra aloe, reddish black in, or brownish black in nature. Its surface is smooth and conchoidal fracture. Then Zanzibar aloe, its color is brown color, dull or waxy smooth appearance. Then next is odor, characteristic odor, then taste intensely bitter and nauseating. Then size, it is found in masses of various sizes. Then solubility, it is entirely soluble in 60% alcohol and partly soluble in water. Next is chemical constant of aloe. We know it is anthracnone glycoside containing drug, so that we need to mention first. Then chief constant is aloin. It is a mixture of glycosides with barbaloin. Then barbaloin is a C glycoside. It also contains isobarbaloin, beta-barbaloin, aloe-imodin, resin, alotic acid, allosin, chrysophenic acid, galactronic acid, choline, saponins, coniferyl alcohol, glucosamines, etc. So this is the structure of aloin. Also contain allosin. Next is chemical test for aloe. So first is general test. Here the 1 gram of aloe powder is boiled with 10 ml of water. Filtrate is used for Brahmin test and borax test. So first one is Brahmin test. Here the small quantity of filtrate is treated with Brahmin solution. It shows some pale yellow precipitate. The next is borax test. Here above filtrate is treated with borax, then shaken well till it dissolves. Then a few drops of this solution are added to a test tube nearly filled with water. So it shows some green fluorescence. Next is some special test or specific test. So first one is nitrous acid test. The crystals of sodium nitrate along with some small quantity of acetic acids are added to aqueous solution of aloe. So different types of aloe shows different observations, like Cape shows faint pink color, Curacao shows sharp pink to carmine color, then Socotra very less change in color, then Zanzibar very less change in color. This test is due to the presence of isobarbaloin present in aloe. Next is nitric acid test. So here treating with nitric acid to the drug or aqueous solution of aloe. So Cape aloe shows brownish to green color. Curacao aloe shows deep brownish red. Socotra aloe shows some pale brownish to yellow color. Zanzibar shows yellowish brown color. In case of cupraloin test, it is also known as Klunk's isobarbaloin test. So here the very little dilute aqueous solution of aloe is treated with saturated copper sulfate solution, then followed by a little quantity of sodium chloride and is excess of 90% alcohol. So in case of Cape aloe, it shows faint coloration rapidly changing to yellow. Curacao aloe, wine red color persisting for 4 hours. Then Socotra and Zanzibar aloe, no color change. Next test is modified anthracnone test, mainly for C glycosides, that is aloe. Then here already we have discussed in previous slide that how to do modified anthracnone chemical test. Again same we need to repeat here. Then finally you will get some pink cherry or pink red color. We have to treat with the ferric chloride solution and organic solvents like chloroform or benzene. Then organic layer we need to separate, and to treat with ammonia solution we'll get some pink to red color. Then uses of aloe, that is purgative mainly. Then in high dose it may act as abortifacient. Some other uses are anti-inflammatory, Then ointment of aloe is used for skin burns, thermal burns, radiation burns, skin irritations. It's also used in hair tonic. Then it's having soothing effect, moisturizing effect, then healing properties. Widely it is used in some cosmetics products, then used for the manufacture of soap, shampoo, sunscreen, moisturizing cream, everything, etc. So that's all about aloe and bitter almond. In this anthracnone glycoside, one more drug we have to discuss, that is Senna. Synonym is Sonamukhi or Acacia senna. Biological source, it consists of dried leaflets of Acacia angustifolia, that is Tinaveli or Tirunelveli senna or Indian senna, and Acacia acutifolia, that is Alexandrian senna or European senna, belong to the family Leguminosae family. Geographical source, mainly it is indigenous to eastern and southern Africa, Europe, India. In India, it is mainly cultivated in Tamil Nadu, Tirunelveli or Tinaveli, Madurai, also in Andhra Pradesh, Gujarat, Rajasthan, etc. Alexandrian senna or European senna, it is cultivated in Africa, Egypt, etc. This is the picture of senna. This is the dried senna leaves. Coming to the macroscopical characters of senna, color is pale green or yellowish green, order none. Taste mucilaginous, bitter, characteristic. Size 7 to 8 millimeter width, 2.5 to 6 millimeter in centimeter in length. The leaves shape, leaves are lanceolate or ovate, entire margin. Apex is mucronate, that is small spine at the top. Then surface, it is glabrous, that is free from hair. Then condition dry. Then type leaflet to peripinnately compound leaf it is. Next is microscopic of senna leaf. In this microscopy, we can divide into mid-drip and lamina portion. So in case of lamina, again divided into upper epidermis, mesophyll cells, lower epidermis. So upper epidermis, it is a single layered polygonal type of cells covered with a thick cuticle. Some cells contain mucilate cells. Then same like upper epidermis, lower epidermis also we can explain same like thing. Then next is mesophyll cells. Mesophyll cell again divided into three portions. One is upper palisade, spongy parenchyma, lower palisade. So the upper palisade, it is a single layered elongated type of cells. They are compactly arranged cells. In case of lower palisade, here it is present only in lamina, not in midrib. But here you can see the upper palisades are continuously in both midrib and both lamina. But one exception in case of lower palisade, lower palisades are occurs only in lamina, not in midrib. So next is spongy parenchymas. These are thin-walled, loosely arranged parenchymatous cells with some intercellular spaces are there. Spherophytes are there. Spherophytes means spherically arranged raphites. That is spherically arranged needle-shaped calcium oxalate crystals. That is spherophytes. So in case of mesophyll cells, we have spherophytes also. Then loosely arranged parenchymatous cells. Next is some in case of lamina we can see some stomata also. The next is midrib portion. So midrib portion again divided into epidermis is there, upper and lower epidermis. All that already we have discussed in lamina portion. Same thing you can mention here also. The next is palisade cells. That is also already we have discussed. That is elongated, compactly arranged palisade cells. Next is crystal sheath. These are parenchymatous layer, contain some calcium oxalate crystals as prism crystals. Then it is present at dorsal and ventral side. Then next is sclerenchymatous sheath below the crystal sheath. So this sheath is lignified cells, thick-walled type of cells covering covering the both xylem and phloem, that is vascular bundles. So the sclerenchymatous sheaths are covering the vascular bundles. So this is the vascular bundles like arc-shaped vascular bundles. So here we have xylem, another portion is phloem. The xylem is lignified and it is present at the ventral side. Then phloem is non-lignified and present at the lower side. The last portion is collenchyma. It is multi-layered thick-walled cells. It contains cellulose. It is present only at ventral side. So this is the transverse section of Senna leaf, two portion, lamina and mid-rib portion. Next is chemical constituents of Senna. So we know it is anthracnone glycoside, contain Sennosides A, B, C, D as chief constituents. Then phytoconstants is responsible for purgative action. Already it is used for purgative action. So this Sennoside ABCD is used for the purgative action. Other constants are renate glycoside, Aloe, palmidin A, camphorol, isoramnatin, then chrysopenic acid, saponins, sterols, mucilage, resin, salicylic acid, etc. So this is the structure of Sennoside C and D, A and B. Again, coming to the chemical test, already we have mentioned Born-Tragers test. Senna main chemical test is Born-Tragers test. Here we have to boil the drug with the sulfuric acid. Filter. To the filtrate, we need to add any organic solvents like benzene or chloroform or carbon tetrachloride. Shake well. The organic layer we need to separate. Then after separation, we need to treat with the ammonia solution. It will form ammoniac layer, shows pink to red color. Then uses, mainly it is used in case of laxative in habitual constipation, that is prolonged constipation. Then purgative in perpetual constipation, that is everlasting constipation. So that's how I conclude this video with three drugs: bitter almond, senna, aloe. Thank you.